Hello students. Today let us discuss about properties of enzymes. This we are discussing from chapter 6 biomolecules part 6. So enzymes basically they are protein in nature. They are made up of chains of amino acids. So all enzymes we can say are protein in nature. They have a three-dimensional nature. That is, they have one or more active sites, and to these active sites, a substrate is binding to form a substrate enzyme complex. They are acting as biocatalysts. They do not participate in the chemical reactions, but they increase or decrease the rate of chemical reactions without actually undergoing chain. A small amount of enzyme can transform a very large quantity of substrate into a product. For example, sucrase is an enzyme which can hydrolyze 1 lakh times of sucrose as compared to its own weight. So this means that even a small quantity of enzyme is capable of transforming a large amount of substrate into product. I hope you understood. So enzymes, protein in nature, biocatalyst having active sites, few or more in number, and they can convert large amount of substrate into products. Let us see further properties of enzymes. They have high turnover rate or high turnover number. For example, the number of substrate acted by 1 micromole of enzyme per minute is quite high. For example, 5 million for catalase. Catalase is an enzyme which participates in conversion of hydrogen peroxide into hydrogen and oxygen. And this catalase, it can convert about 5 million of its substrate. 18 million for urease enzyme. Urease enzyme acts on urea, converting it into ammonia and carbon dioxide. 36 million for carbonic anhydrase. So you can see they are having a high turnover number. Now, if we take carbon dioxide and if we dissolve this carbon dioxide in water in absence of any enzyme, especially carbonic anhydrase, only 200 molecules of carbonic acids will be formed. But if you take the same carbon dioxide and dissolve in same quantity of water in presence of this enzyme carbonic anhydrase, 6 lakhs molecules are formed. That is, you can see that how high turnover these enzymes are having. They increase the speed of chemical reaction and also increase the capacity of the product form. Now, let us see further. Enzymes are specific in catalyzing a reaction. Specific means for specific substrate there is a specific enzyme. Suppose we are talking about urea. Urea will be acted only by urease. If we talk about sucrose, it will be acted by only sucrase. So if we are talking about lactose, it will be acted by only lactase. So enzymes are specific for specific type of substrate. They are sensitive to temperature and pH. At low temperature, enzymes are inactivated, and therefore we are storing all the perishable food items into refrigerator to lower down or to make the enzyme activity inactive. Whereas at high temperature they are killed, they get denatured, and therefore we boil our milk at home and keep it so that it shouldn't be spoiled. They are also affected by pH. Different enzymes can work at different pH. Increase and decrease of pH declines the enzymatic activity. For example, pepsin is a protein digesting enzyme. This can work very well in stomach where the acidic medium is present because of secretion of hydrochloric acid by gastric glands. pH is 2, and only in acidic pH this enzyme pepsin can work. Whereas there is another enzyme trypsin which is secreted in our intestine. This is active at alkaline pH, that is 9.5. So different enzymes, they can work very well at different pH. Enzymes can catalyze reversible reactions also. Substrates are converted to products, and these products can be converted back into substrates. So such type of reaction are also catalyzed. Reversible reactions can be catalyzed by enzymes. And I already told you they are substrate specific. If we heat substances, enzymes are destroyed or denatured. So enzymes are destroyed at higher temperatures and they become inactive at low temperatures. Most enzymes they work at an optimum temperature of 20 to 35 degrees centigrade. So these are all the properties of enzymes. I hope you have understood. In next video, we'll discuss about naming of the enzyme, that is nomenclature. Thank you.